Hey everyone, I'm Roman, and you found the channel where we talk about medicine, science, productivity, and well-being. I'm very proud and excited that my first YouTube video is about something as interesting as what we're gonna talk about today. Something revolutionary happened a couple of weeks ago in science, specifically in chemistry, which has the potential to change the way we view everything. So what is this incredible finding? Well, scientists just observed the first ever stable single-electron covalent bond. In this video, we will explain the discovery in a simple level of understanding and then to a bit more complicated level of understanding for those who want the extra juice of the topic. We will analyze this research as well as how it can aid in future innovations in science and medicine. We will connect the discovery to the bigger picture in science and health. Finally, a conclusion where we wrap up the key points mentioned. Let's start with a simpler version of the story. You have probably heard about carbon, even if you're not a scientist. It is the foundation of all human life. It exists around us. Everything is made of carbon. We are made of carbon. It is what makes us carbon-based life. For this reason, carbon is characterized as the building block of life. Why is that? Carbon has a very special property, which is to form organic bonds using up to 4 electrons. By having this potential of binding, it can create a range of different molecules, ranging from soft molecules to very, very hard molecules, or to organic molecules like the macromolecules we are made of. It creates the soft graphite in your pencil, and it also creates the diamond ring you paid 10K for to give your fiancé.
Oh, you've got a fiancé, you're engaged.
Now, where does this very exciting discovery come into play? This is how bonding works in general, and especially as we knew it before this discovery happened. Atoms have a nucleus made from protons and neutrons, and electrons that work in orbitals around this nucleus. Different atoms have a different number of electrons. For example, oxygen has 8 electrons, and beryllium has 4. For the atom, some of these electrons exist close to the nucleus, and some of these electrons exist in the outermost part, a bit more away from the nucleus. These are the electrons that actually participate when this atom binds with another atom. When two atoms bind together, essentially what happens is an electrostatic interaction between the outermost electrons from one atom with the outermost electrons from the other atom. Bonds come in different types. We have covalent bonds, where two atoms share their outermost electrons, which we call valence electrons. We have ionic bonds, where one atom is so electronegative it kind of steals the electrons from the other atom. And we also have polar bonds, where there is sharing, but one of the atoms pulls the electrons a bit more than the other one. This discovery refers to covalent bonds. There is also metallic bonds, where electrons are able to slide past each other and move around the structure of metallic bonding, but it's not relevant for today's video. So you might say, okay, it seems like we know how bonding works. What's the big deal with this discovery? I'm gonna tell you. Usually when atoms bind, it's like they shake hands, sharing 2 electrons for every bond, 1 from each atom. But this new discovery shows that carbon atoms actually have the potential, under the right circumstances, to hold their hand alone, by having a single electron forming a bond. It is a weaker type of bond, and more fragile to break, but it is a bond.
Bond. James Bond.
To make this happen, scientists had to create an environment where it allowed for this fragile, weak bond to exist, and not get destroyed immediately because of a variety of factors. Let's do our more in-depth analysis before we move to how important is this research actually. The concept of a single-electron carbon bond has existed as a topic in theory for about 100 years, first proposed by Pauling in 1931. Since Pauling's time, scientists have observed single-electron bonds in other substances, like metals, but it's not as important because in metals, electrons are able to continuously move around. Unlike carbon, where in organic molecules, there needs to be a stable bond for the molecule to exist. Feel free to skip that part if you're bored about the details of the actual in-depth science. So how did scientists make this possible in 2024? couple months ago. Researchers from Hokkaido University finally confirmed that single carbon-electron bonds can actually happen. The team used a modified HP derivative, which under normal conditions would go ahead with two-electron oxidation, but they manipulated this structure to perform single-electron oxidation. In this way, they were able to stabilize the resulting radical cation in a way that allowed them to isolate it. The key innovation here was creating a rigid molecular structure with a stretched-out carbon-to-carbon bond, 1.8 Armstrong instead of 1.54. For this, they used an asanaphthalene bridging unit. This elongated bond was crucial because it lowered the electron density between carbon atoms, allowing a single electron to be shared instead of two. Using advanced chemical techniques like X-ray crystallography, scientists were able to confirm the presence of this single electron bond in experimentation, a technique that didn't exist in Pauling's time about a century ago. Normally in a covalent bond, two atoms share a pair of electrons, creating what we call a sigma bond. However, in this case, we're dealing with a single electron bond. Its properties is that it's much weaker, but also much more reactive, because it craves another electron, which means it can form a variety of bonds much easier than under normal circumstances. The electron sharing, in detail analyzed by quantum chemistry, happens by the overlap of molecular orbitals of the carbon atoms. Think of the orbital as a sphere of high electron density. When two orbitals overlap, there are interactions which create a bond. The single electron bond also reflects the quantum nature of electrons. The implications are vast, and it could lead to very serious innovations in nanotechnology, spintronics, and potentially even new materials that harness the power from this quantum behavior for more specific advanced applications. Now let's talk about the importance of this research and potential applications, specifically in medicine, healthcare, and biosciences, since it is mostly what we talk about in this YouTube channel. At its core, this new finding completely changes the way we understand chemical bonding, starting from high school, where we first learn about the difference of covalent, ionic, and metallic bonds. If a couple months ago you said to your chemistry teacher that a covalent atom could have a single electron covalent bond, unless he was super informed about Pauling's theory, he would probably laugh at you. As previously mentioned, carbon forms the foundation of life, and a change in the way it forms bonds could change the way we see things in total. It could open up entirely new avenues for studying the biomolecular interactions of living systems. In biochemistry, carbon is important because it is the core of all chemical interactions that lead to the macromolecules we have in our bodies. From the structure of DNA, to proteins, to nucleic acids, to the foods we eat to get energy from, they are all carbon-based. I cannot highlight this enough. A deeper understanding of such fragile single-electron covalent bonds could help us model how transient states occur within the body. For example, this discovery could influence how we view reactive oxygen species, essentially unstable oxygen molecules that vibrate close to our cells and destroying them, which occur by a variety of factors like radiation, smoking, and, in general, a bad lifestyle. If we can grasp how single-electron bonds occur within a controlled experimental environment, we might also be able to understand short-lived bonds in our body and how they aid in dynamic molecular changes in cells. These interactions could influence anything from enzyme functions to genetic regulations. Second, pharmacology and drug design. In pharmacology, this discovery could reshape the way we view drug development. Drugs function by interacting with biomolecules in our bodies, often binding to specific receptors or enzymes. Traditionally, drug molecules are designed based on chemical bonding. But the existence of single-electron covalent bonds in organic molecules could open up the possibility of designing drugs that target previously unreachable transient states in biomolecular interactions. For example, this could be revolutionary in the study of drug metabolism. When we introduce drugs into the body, they go through complex biotransformations, often involving the formation and breakdown of radicals. A better understanding of these electron bonds could help us design drugs that more efficiently target these radicals, leading to more effective treatments and with fewer side effects. Additionally, this finding could also have implications in designing nanoparticles or delivery systems that target these fragile bonds to release drugs in a controlled manner. Imagine drugs that can be precisely controlled and manipulated based on this quantum state, which we previously had no idea about. It was a hypothesis, but now we know it can actually exist in real life. Let's talk about understanding the body's molecular machinery. Our body works primarily in mechanisms, out of which we know some, but not all. Think of mechanisms involved in things like muscle hypertrophy when you work out in the gym, mechanisms like digestion, or mechanisms that exist in our brain when we talk about neuroscience. If some of these mechanisms incorporate this single covalent electron bonding, which we are now familiar with, there could be a possibility that we discover some of these mechanisms further, which we previously hadn't known, perhaps because we couldn't examine them if they existed in a state which we didn't target for when doing experimentation. It could help us explore, for example, how membrane-bound proteins, for example in cell membranes, work as enzymes or as receptors, especially in transient, highly reactive states. This could lead in innovations to how we treat diseases that stem from membrane dysfunctions, such as neurological disorders or metabolic diseases. Finally, this discovery expands the theoretical framework of how molecules interact within cells. Many biological reactions involve temporary highly reactive states. For example, enzyme-substrate interactions, where bonds literally break within nanoseconds. Being able to study these bonds with single electrons in biological living systems could give us insights into fundamental reactions that before now could potentially be happening under the hood unnoticed. It could enhance our understanding about processes like cell respiration, protein synthesis, and even immune response, each of which depend on precise timing molecular events. In short, while this breakthrough might seem too theoretical, its practical implications in the field of medicine and health sciences could be enormous, potentially. With this, I want to thank you for paying the attention you did, and by staying here until now, the end of the video, I probably guess that you're interested in science, medicine, and perhaps some of the other things we're going to talk about in this channel, which include productivity, wellness, and more. So perhaps you should consider subscribing so you don't miss any of the future videos. What are your thoughts about this scientific finding? Write it down in the comments.